Today we're discussing Chapter 5, Section 2, which covers the topics of mitosis and cytokinesis. To begin our discussion, we need to talk about the genetic material of the cell, which is found in the chromosomes. A lot of what's happening in mitosis has to do with the chromosome movement. So at the beginning of mitosis, the DNA actually has to wrap itself around special proteins called histones in order to form that highly condensed structure that we know as a chromosome. So this is what it looks like here. This X-shaped chromosome is probably what you're familiar with seeing, and if you were to see that picture, you would immediately recognize that as a chromosome. But what it is the DNA double helix has actually wrapped around these histone proteins, and then that thread of DNA and protein coils even again to form this chromatin, and the chromatin is the normal state where we find the DNA in the cell. The chromatin is relatively relaxed. What this structure is made out of is that supercoiled DNA. So this spiral is that chromatin thread spiraling and condensing and winding up even tighter than normal. When the DNA is wound up tight like this, you can't really access it to express genes. In the chromatin stage, the DNA is loosely wrapped, and so it's easy for the enzymes to access that DNA, and they can transcribe it and then go on, and translation can happen at the ribosomes. But in this supercoiled state, that DNA is wound up so tight that it can't be expressed. And so this state is what makes a chromosome looks like this X right here. And this only happens during cell division for the purpose of keeping that DNA nice and compact so that it can be moved without breaking it. So let's take a look at the parts of a chromosome. There's a lot of vocabulary that has to do with chromosomes, and unfortunately it mostly starts with a C, so it can be a little bit confusing. On the right, you see a picture of that duplicated chromosome. There's two sides of the X, and we'll talk about what it looks like if it's duplicated versus not duplicated. There's a picture on the next slide. What we're looking at here is a supercoiled version. So we're not looking at the chromatin. Remember that chromatin was the relaxed thread. When the chromatin condenses, then we have a chromosome. So definitely know the difference between those two words. Both of these structures are made out of DNA that's wrapped around histones. The only difference between them is that chromatin is more relaxed and the chromosome is the exact same material, it's just more tightly wrapped. So we're not seeing a picture of chromatin in this picture off here to the right. This is the supercoiled chromosome structure. I will show you some chromatin a little bit later in this presentation, but it really just looks like threads or even a pile of spaghetti. Now this structure here is that highly condensed chromosome, and there are two identical pieces here. The box on the left is showing you one intact piece of DNA. Basically, every gene that belongs on this chromosome is on this half of the DNA. And then the other half of the chromosome here is an exact genetic copy. So that's why we call this a duplicated chromosome. If you are doing cell division, it makes sense. You're going to need two copies of all of your DNA. So this was the original copy, and this would be one chromatid. And then after DNA replication, after the S phase, all of the DNA gets copied. So there's a second identical chromatid, and your cell keeps those chromatids organized by joining the identical ones together at the center, which is called the centromere. That's the point of attachment. So this is considered one chromosome, but it's a chromosome after DNA replication. So the parts of this chromosome would be two identical sister chromatids. There was the original chromatid, and that got duplicated during the S phase. So now there is a second identical chromatid, and they are attached at the centromere region. So this diagram does a good job of explaining what a chromosome looks like, because a chromosome will look different depending on what stage of the cell cycle we're in. If the DNA has not replicated, your chromosome would just look like a single rod, like what we see in this first picture here. DNA hasn't copied itself. There is no duplicated chromosome. There's no identical sister chromatid to attach here. So the chromosome just looks like one single rod. But after DNA replication, when the S phase is over, that initial rod has been copied, and now there is a second one, so that's why we have our two identical chromatids attached. We still call this thing one chromosome because they're literally attached right here in the center. It's still one piece. If I were to tug on one corner of this chromosome and pull it over to one side of the screen, this whole X would move. And so that's how I know it's one chromosome, because it's all attached together in a single piece. Now we'll talk about mitosis in the rest of this presentation and how this process happens, but essentially the purpose of mitosis is to split those sister chromatids from each other. So at the end of mitosis, you're going to end up again with that single rod chromosome. It's no longer a duplicated chromosome because we have two different daughter cells. Each one is going to get half of this X. So we're back exactly where we started at the end of mitosis. So hopefully that helps to clarify a little bit about chromosome structure and the vocabulary. I understand that it can get complex, so if you have any questions about that or if you need extra practice, please just let me know and we can certainly go over it again. So now let's talk about the actual stages of mitosis itself. We talked earlier about the cell cycle, and the M phase of the cell cycle is when mitosis occurs. The process of mitosis is simply the division of the cell's nucleus. And we are going to be focusing just on what happens with the genetic material in the nucleus, and then also these centrioles here, if this is an animal cell, their role in manipulating the chromosomes inside of the nucleus. So mitosis is just a eukaryotic cell process. It is specific to the nucleus. It is specific to structures that are found in eukaryotic cells to help divide that genetic material. So in interphase, we learned during the cell cycle that interphase is when all of the preparation happens. The cell is growing, it's doing its normal function. Eventually, if it is destined to divide, it will copy its DNA and it will produce all the proteins needed for cell division. So after interphase, if everything looks good, then the cell will proceed into mitosis. Our first phase of mitosis is called prophase. There are a few things that happen in prophase. The chromatin, which was that thread on the previous picture, the DNA did not look like chromosomes like this, right? It looked like long strings or threads. So it was in the chromatin form during interphase. Now that we have started prophase, one of the very first things that happens is the chromosomes are going to supercoil and they're going to condense into these X shapes. And again, they're X-shaped because we have already finished the S phase. So DNA has already been duplicated, so when those chromosomes condense, they look like X's. Now we're going to be dividing chromosomes and separating them from each other, so we're also going to have the breakdown of the nuclear membrane. You can see that in this picture here because of the dashed line. So we're getting rid of that membrane, breaking it down in order to be able to move the chromosomes. And in order to move the chromosomes, the structure that does that would be the spindle fibers. You can see the spindle fibers are these lines right here that are forming off to the side of the cell. Now, this is an animal cell, so animal cells have in them, centrioles. That's what these structures are. Plant cells don't have this exact arrangement of centrioles. They do still have the ability to make spindle fibers. You just would not see those centriole structures if we were looking at a plant cell here. All right, our next phase of mitosis is metaphase. This is probably the easiest one to recognize. At this point in time, notice how the spindle fibers are much longer, and what they have done is they have attached to the centromeres of those chromosomes, and it's the spindle fibers that actually push and pull on the chromosomes to move them into the correct location. So by the time all of the chromosomes line up in this single file line in the middle of the cell, we have reached metaphase of the cell cycle. And this is really a key phase because it's how the cell is able to determine if it has organized all of its chromosomes correctly. All right, next we have anaphase. And in anaphase, notice that those chromosomes are now being pulled apart. The spindle fibers shorten, and that pulls on each X. It takes one half of the X to one side of the cell. The other half of the X is going to the other side of the cell. So we used to have all 4 of these chromosomes lined up in a single file line in the middle of the cell, but now we have each chromosome is being split in half. Half of it is going over here, half of it's going over here. Remember, these were identical sister chromatids. So this information, whatever the DNA sequence is on this chromosome, is exactly the same as the one on the right. Same for the red, yellow, and blue chromosomes. At this point, we no longer have X-shaped chromosomes because we've separated the copies from each other. And now each individual chromosome would just look like a single rod. Our last stage of mitosis is going to be telophase. Telophase is pretty much exactly the reverse of prophase. In telophase, we have 2 new nuclei that need to form. So in prophase, remember, we had to get rid of the nuclear membrane. Well, in telophase, we have to build it back. And you can see that process happening here. This dark purple nuclear membrane is starting to form again around both of those nuclei. Spindle fibers are going to finish breaking down. We don't see very many spindle fibers left at this point. They're not really needed anymore because we've already moved the chromosomes, and the function of the spindle fibers is to move chromosomes. And then finally, the DNA is going to relax back into its chromatin state. That's not really shown here. This is not chromatin yet. This is still showing condensed chromosomes, but it will be in the process of starting to relax. Pretty much as that nuclear membrane finishes forming, that's when the relaxation can start occurring. Now you'll notice this overall cell shape here has this pinching in the middle. That is going to be the beginning of the next stage, which is cytokinesis. So a lot of times there's an overlap between telophase, which just describes what's happening in the nucleus, and cytokinesis, which describes that pinching in of the cytoplasm. So let's talk about cytokinesis. This is the splitting of the cytoplasm. In an animal cell, there is a ring of proteins called a contractile ring that will basically get smaller and smaller until it pinches off the cell in the middle. And you will notice here, now that telophase is finished completely, we've gone back to having a complete nucleus in each cell, and we have that relaxed thread-like chromatin in each nucleus instead of the supercoiled chromosomes. Now, in a plant cell, it's a little bit different. Remember that plant cells have a cell wall outside of their cell membrane. So they basically need to build a brand new cell wall in between these 2 daughter cells. And they do that by depositing vesicles filled with cell wall material. And it's basically just like building a wall between the 2 cells. So in an animal cell, we just pinch off the cytoplasm, and in a plant cell, we build that new cell wall between the 2 daughter cells. As it's forming, it's called a cell plate, and then once it is complete, we call it a cell wall. So that's the basics of mitosis and cytokinesis. You will notice on the back of your Power Notes sheet, you have some practice questions, a few vocabulary terms you can practice with, and then you can sketch those phases to make sure you understand them, and then some questions at the end. So I encourage you to do this as you're studying, and if there's anything on here that you have a question about, please ask me in class.